So welcome back, everyone. This part of the talk is going to be a little more sort of getting into some of the details of some more practical issues. I'm going to talk first about using existing ChromHMM annotations. So in this case, this is sort of using as resources generated from previous work. And then the second part of the talk, I'll talk about if you wanted to actually use the software on some of your own data. And I'm going to talk about how to access some of the ChromHMM annotations from the browser, and then also talk about some examples of some snip-focused databases. So these are databases of single nucleotide polymorphisms, which are have incorporated some of these annotations to help interpret SNPs. So I'm going to give you talk about some of the major models that I've published or collaborated in the publication across multiple human cell types. I just want to make the point there's other models out there. Other people have published them in specific systems and in mouse, and also there's been some other consortium projects that have used this that I haven't specifically been involved with, but I'm just going to give you some of the major ones that I've been involved with, and to sort of show you how you could actually go about accessing them, and sort of mention some of the differences between the models. So this was the original model across multiple different cell types, which I was using before. So this was the work published in 2011, where it was based on 9 marks, or 8 histone modifications in CDCF, as well as an input control across 9 cell types. And this is incorporated into the UCSC genome browser. So this is the URL if you haven't been there before. And if you go there, then you click Genome Browser. And then under the regulation section, so in the browser, you would scroll down, and then they have these different categories. So it would be under regulation. There's this window, encode histone modifications, and then you would want to change this to show. And then you would also follow this hyperlink. And then there's going to be a set of 3 different histone sets from different groups. If you wanted to see the raw histone signal that went into this, you would click the broad histone. But I'm going to show you here the breadcrumb, which tries to summarize these multiple histone modifications. So then you would click here, and then you would get to see this window here. And then you would select the specific cell types you're interested in. And you can play with the mode here in terms of dense means you just get one single line per cell type. If you expanded it out, you could get potentially different state calls in different cell types. I tend to like it in dense mode, except sometimes I've colored 2 states the same color that are similar, and if you wanted to really differentiate them, you might need to change the mode to see that. And then you can select the specific cell types you're interested in. If you wanted all the cell types, you would click the plus sign to get them all. And then this would show you the list of tracks you're selecting, those that are sort of in dark. One thing to note here is that there's a downloads button. So if you didn't want to just display in the browser, but you wanted to download the annotations in a standard bed format, you would click on the downloads link, and then it would give you a menu where you can select which files to download. And that's sort of going to be generally true for other ways I'm going to access this. And then they have a description of this, and it has sort of our candidate annotations and the coloring scheme right there. And this is showing you, I've just pasted in those emission parameters that are corresponding to this that I showed you before. So then once you have selected the cell types you're interested in, if you go back to the browser window, what you'll now see is these annotations appearing in the browser. And then each row is corresponding to a different cell type. And then you see the specific chromatostate annotations for that 9 cell type model. The next model, so this work was done specifically in the context of the ENCODE production group at Brad Bernstein. We also did an integrative model, which in this case, it leveraged the same set of histone modifications and CDCF, but we also combined it with RNA pull2, so this is the RNA polymerase ChIP-seq. And then, as I was talking before in response to a question in the earlier session, DNase assays. So these are assays which measure the open chromatin. And there's two different protocols for measuring the open chromatin, one by a group at the University of Washington and one by a group at Duke. And they have overlapping, but in some cases different properties. And then FAIR, which is another protocol for measuring open chromatin. So it's a little deeper in terms of the mark coverage, but it was on 6 of the 9 cell types. And this is under, this was the main publication, and then it was also described or mentioned in the main ENCODE consortium publication and some additional analysis in this 2013 paper. So in this case, these specific segmentations are, if you go under genome segments under the regulation tab, and then what you're going to see here is this menu here, and then it has the 6 cell types, and then ChromHMM is sort of the primary one I'm focused on. There's another alternative method, Segue, by a group at the University of Washington. And then we had a simplified attempt at merging the two. But in general, I tend to use the ChromHMM segmentations. But just sort of letting you know that that's going to be available. And then if you scroll down on that page, similar to before, you're going to see the different rows correspond to the different states, and here are the different marks of the mission parameters. I've pasted that in here, and then these are the different characterizations of these states. And it's talked more about in the references I had given you, these different states. And then similar to before, if you go back to the browser, then you can pull out these 6 cell types in their chromatin state annotation. And it's roughly similar color coding as before, with like the green is the transcribed regions, gray is more repressive, yellow is enhancers, red is promoters, blue is insulators. Now, the last 3 sets of models were all sort of published in the context of this Roadmap Epigenomics Consortium work earlier this year. So there was 127 different samples. They're not all cell types. Sometimes they're regions, like a region of the brain. And sometimes we had multiple different individuals as a separate epigenome. But in total, we had what we term like 127 reference epigenomes. That we had 5 marks directly based on the observed data. There was an auxiliary model based on the observed data where we added H3K27 acetylation, but then that dropped us to 98 cell types. And then, as I had talked about at the end, with the imputed data, we took the 12 marks here that I've listed, including DNase and then these histone modifications. H2AZ is a specific histone variant, and learned a chromatin state model, in this case with 25 states based on that data. And this was published as part of this publication and then also referenced as part of the main Roadmap Consortium work. So how to access this one through the UCSC browser, there's an additional step because it's not on the main page. You have to use something called the Track Hubs if you want to access it if you're going through UCSC. So Track Hubs, if you click right on this button here, what it'll bring you is a menu. And then the one I'm going to show you here is this Roadmap Epigenomics Integrative Analysis Hub, and then you would click Connect. I noted there's going to be another Track Hub which will sound similar, which is the Roadmap Epigenomics Data Complete Collection, which has more of sort of the primary data without as much, not completely matching sort of the integrative analysis where a lot of the data was reprocessed and consolidated. So if you're going to sort of go to this site later, just sort of be aware that there's two different views on the roadmap data, and I'm focusing here on the one that's called the Roadmap Epigenomics Integrative Analysis Hub. And then you would click Connect, and then if you go back to the main browser window, what you're going to see now is these 5 different options to come up. What CON means is it means consolidated. So what the consolidated samples refer to, these are the 127 reference epigenomes. In some cases, if we had multiple different experiments for a mark in some cell type, we had combined them often in order to get sufficient sequencing depth. Unconsolidated was sort of the raw experiments where we didn't pool any library together. And then you can either do it by assay, by the biological sample, or specific identifier. And I'm going to focus here on this by assay menu to bring up the chromatin state annotations. So the first thing that'll come up is here this ChromHMM track. So what you're going to want to do is change this one to, so here you would want to change this to show, and then here you would want to change this to dense. And then there's going to be, on that page, 3 different models. So these are the 3 different models. The primary one is the 15-state model based on 5 marks. This is the one based on K27 acetyl. And then this is the one based on the imputed data, which I had mentioned here based on the 12 marks. So those will all be on this page. I've had to copy them there because you can't view them all at once on a screenshot. So then when you go to this page, there's going to be, so auxiliary is the one with H3K27 acetyl. Primary is the one based on the 5 marks. These 2 are based on observed data. And then the imputed data one is the one based on the 12 marks on the imputed data. And then, so if we were, so there's 2 sort of sets of boxes here. One is the data type. So you would click, you would need to check if you were interested in observed data, or the, what's called real, and then the imputed data, or just one of these. Though it's the case if you had only checked, for example, real, and then you checked imputed, then you wouldn't get anything here because there wouldn't be anything that matched the criteria. So there's some redundancy in these boxes or conflicts. But in this case, what you can do is just always have them both checked and then just control it over here. So what this is saying, what I've done is I click the plus here. So what I'm getting is the imputed chromatin state model across all the cell types for which it's defined. If we were interested in the primary model based on observed data, we would check the plus here, and if we wanted the auxiliary, we would check those, and then probably you would want to only look at one of these models at a time and uncheck the others. So then if you go back now to the browser, it's going to be much deeper in terms of the cell coverage. This is showing you the imputed model, and it's not all the cell types, it's getting cut off. So then you can see, based on the imputed model, what the chromatin state calls are. So that is if you're going through the UCSC genome browser. The roadmap epigenomics data, chromatin state annotations can also, a lot of the data can more directly be downloaded straight from the portal that was created as part of the supplementary website with the main Nature paper, which has sort of a clearinghouse for all the sort of consortium integrative analysis data. So there you can get sort of the raw processed data, also the signal tracks with imputed data, and then these chromatin state annotations based on the core model, this 18-state model, and this imputed model. So if we clicked, if we go, for example, to this core 15 model, what we'll get is this page here. And then it has links to sort of some raw downloads of the files related to it. And then there's also this option here to visualize. And then you can either click this button, this check mark, to open a new page, or if you unclick it, it'll open in the same page, and it'll be a visualization. And I'm going to show you when we check it to open in a new page. And one thing you need to be aware of is a lot of browsers will think it's a pop-up, so you need to deactivate your pop-up blocking for this specific site when it asks. And then what opens is a view on the Human Epigenome browser at Washington University. So Ting Wang's group at Washington University in St. Louis has created a very nice resource of a browser sort of targeted at epigenomic data that's integrated with all the roadmap epigenomics data in the chromatin states and has a lot of various visualization and filtering features. So going through the roadmap portal site and then clicking the visualize is probably the most direct way to sort of get the relevant roadmap data you're interested in, but you could also go directly to the Epigenome browser, and then there's ways to pull in the roadmap data as well. I won't go sort of through that in detail, but if that's a resource to be aware of as well for working with epigenomic data in a browser. So that is sort of how to access the raw data, download it, and view it in the browser. Any questions at this point? Yeah.
Yeah, I mean, I assume in practice one should always use the state model based on your imputed data as well, right? For these 127 cell lines, because it's most comprehensive. So could you just discuss which kind of track or states people should use?
Yeah, I mean, so I tend to prefer that model in the sense that it has more marks and it can, in some sense, capture, sort of be more consistent between the cell types because it gets rid of a lot of the confounders. But I mean, with the imputed data, and one thing to be aware of is if you had like some mark which was only observed in one cell type that doesn't correlate with any other marks, then there could be no basis potentially for imputing that. So it can potentially miss some cell type specific behavior. So if you if you have some very complex regulation that doesn't fit with the normal patterns, then the imputed data in those locations could be worse. But for a lot of analyses I've done, have found the imputed data model to be have advantages. But it's sort of a much newer thing, and so it's out there for the community to sort of potentially compare both on whatever analysis tasks they're interested in, and then sort of start seeing what is more useful for their specific tasks.
And could you describe when people should use the cell line data as opposed to the primary cells? Could you give us some situations where you would choose one over the other?
Yeah, one thing I didn't sort of go into detail about, but of these 127 from the roadmap, actually 16 of them were generated by the ENCODE project, but they had been reprocessed using the same data processing pipeline as the roadmap data to compare to. So in general, I would just sort of recommend you use all 127. You're going to get both the primary and the cell line data. I mean, I think a lot of the reason for, the cell lines originally was they were easier to work with, and especially if you were comparing with transcription factor binding data, there's much less of that in primary cell types. But you can get a lot more potentially biologically relevant cell types from the roadmap data.
So the question is, different experiments can have very different signal-to-noise ratios. So you may have like the number of hits you call for certain marks could vary very dramatic on some samples. How does that affect the result of
So this is variability because of technical reasons you're referring to. Yeah, I mean, so there are options that, I mean, you could, if you have like an input control for the experiment, then you could potentially control for some sort of peaks by doing the sort of thresholding based on the input. But in general, I mean, that's one of the arguments for the imputed data, is that if you're relying on any one experiment, there's a lot of variability. I mean, ideally, you would want to do every experiment in a lot of replicates, but that's not feasible often in terms of the sample or the budget. And then, so the imputed data in some sense allows you to both explore more biological space, have coverage of it, but also have some of the robustness properties as if you did a lot of replicates and getting rid of some of the unwanted variability. But I mean, that's something that, I mean, it's an important thing to keep in mind, is that when you're comparing two samples and you see differences, is that differences exceeding what would be expected just for the variability for technical reasons. So that's something to keep in mind. Okay, other questions? So the next part, I'm going to talk about some SNP focused databases. And I'm going to talk about sort of two prominent ones, but there's, I think, other resources out there that have integrated these annotations into allowing one to query it. So I'm actually going to try a online demo of this tool. So this was developed by Luke Ward and Manolis Kellis, and it's called HaploReg. And the idea here is that you can specify, if you're interested in ENCODE or roadmap epigenomes as part of the options, and also some of the populations you're interested in for linkage disequilibrium. And the idea of this tool is that you specify a SNP or a set of SNPs you're interested in, and then it queries the linkage disequilibrium information, such as from the 1,000 genomes, and finds all the SNPs that are in tight linkage disequilibrium with that SNP, and then gives you a table for that SNP and all the SNPs in linkage disequilibrium, what types of annotations it's overlapping. So it has the promoter states and then the enhancer states, and it's telling you what cell types you have that annotation covering those SNPs. And then you have other types of information as well that aren't directly the chromatin states, the DNase data, and whether it disrupts a known regulatory motif. And I'll give you a demo of this tool. So if we go to set options, I've set the default, and I'm going to have it do the roadmap data, which will include the larger set of data. The ENCODE was focused on the 9 cell types. And then I'll use the default settings for other things. And then you can either specify a specific SNP, or it has preloaded gWAS. So I'm going to ask here for the 3 SNPs that were associated with acute lymphoblastic leukemia, childhood leukemia, and then I'm going to do a submit query. And then what it tells me here, it did an enhancer enrichment analysis, and it pulls out CD4-related cells. So this is immune-related cells being enriched here, that 2 of the 3 SNPs were falling into these locations. So it turned out to be significant here, and this makes biological sense. And then one can go into the specific SNPs, and then you see the other SNPs in LD, and then if you mouse over it, you get a list of the abbreviations for the specific cell types which had the enhancer in that SNP. And this is another SNP which had a more focused set of cell types that were relevant.
Could you, when you mouse over it and you see some
Yeah, so here's the abbreviation, I think, for the that were coming up. So like this one, if it's ccc.treg-p, it's CD4 plus, CD25 plus, CD127-treg primary cells.
So over all enhancers and some of the enhancers, so can you read like one line, what does this mean, like the p-value and the other stuff?
Yeah, so this is, so I didn't write this tool, so I might be saying things incorrectly, but what I'm surmising is that it's showing you the number, so the first set is all enhancers, and then the strong enhancers are those that were more state associated with, I think, greater acetylation. And then this is showing you how many were observed, how many were expected, what's the fold enrichment, and a p-value calculation. And then if you restricted it to a different set, a more restricted set of enhancers, the same analysis. And then this is based on the DNase data, which is directly this open chromatin assay, not the chromatin states. And then if you go over here to one of these lines, what it's telling you, the first thing is the chromosome, the specific position. Then this is the query SNP, so that might be the one curated into the gWAS catalog. But then it's telling you other SNPs that are in linkage disequilibrium and what's the LD. And then it's telling you here what's the reference allele, what's the alternative allele, what's the frequency in different populations of each allele, whether that base is conserved based on conserved element set, whether it overlaps promoter or enhancer associated histone marks or states, then what cell types there's DNase in, specific transcription factor binding, eQTL information, which is quantitative trait loci, what motifs for transcription factors might be created or disrupted, and then relationship to annotated genes, and if there's a functional annotation, DB SNP.
And could you just discuss a bit, what should people focus on? Would it be more relevant to look at promoter histone marks or enhancer marks in order to find the relevant cell type? Do you have any opinion?
Yeah, and so the enhancers tend to be more cell type specific, so you can tend to get more relevant cell types often from the enhancers. There are some methods now that people have been developing given a set of various annotations and SNPs that'll sort of score what's the relative importance of different annotations and different cell types as well. So there's some formal ways that, in addition, this is often giving people use as sort of a hypothesis tool. It's not necessarily giving you an explicit prioritization always, yeah.
Is it possible to specify genes of interest, or you have to extract the chromosome regions yourself?
So in this tool, you specify SNPs. It's based on SNPs. And then it'll use the population data to expand that out to other SNPs based on linkage disequilibrium.
Is it possible to input a SNP, but the enhancer histone mark is missing, but no result? But I just input SNP, but those colors are missing.
It could mean that there wasn't any enhancer in any cell type at that position. And also, if you're doing the default encode, then it'll have fewer cell types. If you go to the roadmap, you're going to get more cell types. That was in the set options. But yeah, I mean, there will be locations that don't have any activity at all.
Could you describe what would be a home run? Like what would be a convincing finding based on the output? So for example, some SNPs that change motifs, and then what would be an exciting p-value?
Yeah.
How would you interpret these findings?
I mean, what you would also want to see is that like some SNP would be, if you had a lot of SNPs in LD, that there's only one SNP which was getting a lot of annotations and then the other SNPs weren't, then you could potentially have a lot more evidence that this is the more likely causal SNP. But yeah, so the next tool I'll tell you about, RegularMDB, has a formal scoring system for these annotations based on heuristic rules. And then there's other people who've been developing methods, sort of machine learning methods, to predict the relative prioritization of various annotations on what's more important than others. But I mean, intuitively, you would want to look for, SNPs overlapping potential regulatory sites or disrupting motifs. And I mean, where you get a lot of information from this is also if you have a lot of different SNPs, then you could potentially suggest relevant cell types. And what I've been talking about so far is just based on the lead SNPs, but people have also been noticing that, or the genome-wide significance SNPs, if you go past the genome-wide significance threshold, a lot of the things below that are still potentially real. You just don't have enough statistical power. So you'll have some false discoveries as you go further, but if you're doing enrichment type analysis, comparing the things below the genome-wide significance set against other annotations, you could potentially get more power to start identifying relevant annotations or cell types as well. So this is another tool out there. This is RegularMDB, developed by Mike Snyder's group at Stanford. And this one is set where you can provide a set of SNPs, and then it'll give you some scores based on the regulation, various rules of what type of regulatory annotations there are. And it turns out to also just be a convenient way to pull up all the ChromHMM annotations based on the roadmap model. It's showing you what the coordinates of a certain segment it overlaps, the characterization, what's the high-level grouping of it, and then the specific cell type from it. And then when you do this, you'll also get other annotations of that SNP that is off the screen that I'm not showing you. So that's sort of concludes the part on existing annotations where you wouldn't need to run the software, just sort of using resources out there. I'm going to now move into talking about how, if you had your own data or you wanted to download data and apply the ChromHMM software, how you would go about doing this. So you would go to this website. Here's the website for ChromHMM, and then it's publicly available. You would just sort of click this download link, and it would be a zip file, and then you would unzip it. And then just want to point out, so that's the software download. There's also a manual and PDF that gives all the options. And then just point out here, there's a paper in 2012 giving some more of the technical information about some of the software, and it's mainly in the supplement of that. The overall modeling approach had been described earlier. And then there's a mailing list here. Google announced to get new announcements for new versions of the software, and I'm planning a new version at some point in the near future. So the input to ChromHMM, as I had talked about before, is this binarization of marks at these 200 base pair intervals. So the main command, and I'll sort of illustrate this on the command line, is a command in ChromHMM learn model, which assumes you have binarized data. And then the primary way to get the binarized data is if you have a set of bed files. So if you work with genomic data, you've probably come across bed files. So these have the coordinate, the chromosome, the star at the end, and then has other information, including the strand of the read. It's often, sometimes people have BAM files. That's becoming more common nowadays. And there's ways to convert BAM to BED pretty directly. BED Tools is a primary one, has a program BAM to BED, which you could download and convert your BAM files into BED files if they're not already, if you don't already have BED files of your reads. And then so if you have BED files, you run this command binarize BED, which I'm going to go into details about. So the main thing is you need to have, of all your reads, we're going to assume you have BED files of them. There's other ways to form this binarized data, but I'm going to assume we're working off of aligned reads. So this is, yes?
So we can reach all these tech align file for the 127 epigenome individual epigenomes from proteins in right?
The tag, you're asking about the tag align format? Or the?
You recently published a paper 2 months ago from the individual epigenomes.
Right.
127 epigenomes. So I was looking for the protein script. We can access these files, but they are not in the BED format. So what is the difference between tech align BED format? I think Andrew Leisure was doing this. So what is the step? Because you are saying the BED file and then converting the BED file for the chromosome coordinates, but for their website they have tech align format. So what's the, how can we convert the tech align format to BED format? Or is it the, I can see that the chromosome stack.
Yeah, so I think you can use tag-align similar to as you would use BED. I can't remember off the top of my head what column everything's in, but within, you can use the binarized BED, can be applied to tag-align formats, I think, but you would, I would need to check the exact columns, but we can talk about it offline, if there's any differences, but I think it should apply. What I'm going to talk about should pretty much apply to that as well. There might be a slight difference. So this is assuming also you're going to need to have Java installed on your computer. And so this is a zoomed-in view of the command line, so sort of dissecting this for the binarized BED. The first part is going to be applying to all the commands. So you type Java, and then this is a memory flag. So it tells Java how much memory it has access to. So this is saying 1,600 megabytes. If you're working with some bigger files, you're probably going to need to increase this. And then -jar, and then ChromHMM -jar. So it's telling it to execute this jar file. Then so ChromHMM has a number of different high-level commands. So I'm going to talk in depth about binarized BED and then learn model, and then I'll have some other specific commands. So then you have here, the next thing is, the first parameter to binarized BED is the name of a file with the chromosome lengths. And included with the ChromHMM download is the lengths of a number of the standard assemblies. So then you would, in that directory, just specify which assembly you want. And if the assembly you're interested in isn't in the ChromHMM download, there's a utility that's explained in the manual that UCSC provides, a fetch chromosome size command, which you just sort of run that executable with the name of the assembly, and it'll download that chromosome length file. Then a directory with the BED files, so these are the inputs, and then a file specifying the sort of overall schema of your BED files in terms of what cells and marks they are. So this is telling you, the first column is what cell type it is. The second column is what mark it is, and then this is the actual BED file for that specific cell mark combination is in the third column. And then this last column is optional, but if you want to do your binarization based on an input control relative to that, then you would specify what bed file control has the control files. And I believe you can also put gzips here and keep this zipped as well. And another thing to note with the control is you could also treat it as a mark. Sometimes we do that. If the data tends to be relatively flat, but the input is only peaking at certain like artifactual regions, we tend to put the control as the mark. If we're seeing that the control tends to peak in lots of places at lots of promoters, it's sort of better to binarize relative to that control file and then put it in the fourth column. But it would need to be sequenced reasonably deep to serve as a control. And so in this case, when you have multiple cell types, what it's going to do is learn a common set of chromatin state models, but then learn cell type specific annotations. If you wanted to have multiple cell types, but you just wanted to have one annotation in the genome, you would just give it some common set of cell names and then name the different marks specifying the cell type. And then you could learn sort of a stacked model of all your data at once. And then the last option is a directory name for which the binarized data files are going to be written. So that is how we get the binarized data. Now, this is the main command to learn the model. So the first part of this is as before, and then we've changed to binary. What used to be binarized bed is now learn model. I've put in a default optional, put in an optional parameter here. -P0 specifies to run this on multiple different processors. So if you have a machine with multiple different processors available, with -P0, it can use as many processors as it can get access to, and then it'll run in parallel and run faster. In general, you could have it put an N there to limit up to how many processors it's able to use. And the default is it can just use one processor. So this is to sort of speed things up if you have access to more processors. It does use more memory also as you're using more processors. So if memory is becoming a constraint, you would also want to limit the number of processors. The next file directory is input is the directory which has the binarized input. So this is the output of the binarized bed is now the input to the learn model command. Then you have another parameter here, which is the directory where the output should be written. Then you have a parameter which is the number of states. So often you're going to run this command across a range of different number of states. And I'll show you a command later that allows you to, in one way, compare the different models. And then this specifies the genome assembly that you're using. And ChromHMM has some annotations available to do some automated enrichment analysis afterwards based on if that assembly is found in it. If there aren't any enrichment files matching that assembly, then we'll just sort of ignore that. So after you hit return, it's going to start running, and it'll give you some progress reports. It'll show you sort of the status of the model being generated. And then depending on like exactly how many cell types and states you're running with and how many processors, it could finish in like 10 minutes, or you might need to run it overnight, depending on exactly the scale of your problem. And then it's going to give you a report. So it constructs a website or web page, which summarizes a lot of information and files, and I'll sort of walk you through the output of this. So the first thing is it's giving you sort of a documentation of what the actual command was that was used to generate this report and the specific parameters. So you can have a record of what the command was associated with that specific output. Then the first thing, so I'm sort of making bigger what's shown on the left side, is the mission parameters. So it creates here as default as a sort of a graphic image format. Each row is corresponding to a different state. each column is one of the different input marks, and then the darker blue means a greater probability of seeing that mark in that state. And then if you see below it, there's two links here. One is if you want to get it in an SVG format, so this could be something that could be converted to PDF and used for higher resolution graphics. And then there's another thing which gives you this table, but in a tab-delimited text format, so you can download the actual values of the emission parameters. The next thing that appears is the transition parameter. So this also specifies part of the model. So the rows are the states from, then the state to, and then the relative probability. And similar before, you have the, you can download this as an SVG or also in a tab-delimited text file. And there's also a link here to download the model parameter file. This can be used for other commands later. It's not really designed to be, I mean, it's a text file, but it's not necessarily designed for the user to read this file directly. It's just for ChromHMM to parse if you're going to take this model and you can apply it later in some ways. The next thing that appears are the segmentation files. These are going to be in a 4-column bed format. So what you see here is the chromosome, then the start position, the end position, and then the state. And then the E stands for was ordered by the emission distribution. There's an option to order the states based on the transition structure. And then you see the state number. And then you get a separate file for each of the cell types. So these are more for, not really for the human to read, but if you have some method that wants to do analysis on this, these are useful to have. Now these files are what can be loaded into the browsers. So these are, so it mentions UCSC genome browser, but it's also popular. People use IGV, so this is a downloadable web browser from the Broad Institute. Here's the URL. So you can take one of these files and then you can load it in either through the custom tracks on UCSC, or there's an option here to load the files. And there's two different formats. One is dense, which sort of, you can get things like this. There's also an expanded one which puts each state always on one line. If you wanted to sort of expand it out, I tend to use the dense format. One thing to note is when you run this by automatically, it just sort of picks a color scheming without any real correspondence to what's been used previously. But you can manually decide how to color your states. And then using a command I'll show you later, you can regenerate the browser files based on a different coloring scheme than happens automatically. The next thing, I've scrolled down some, is state enrichments. So within ChromHMM, there's going to be a directory tied to that assembly with various previous annotations. So in this case, or the first column tells you for the states what's the relative frequency of that state in terms of the genome coverage. And then it's showing you in the additional columns the CPG island enrichment for the nuclear lamina, for rough-seq annotated exons, genes, ends of transcripts, transcription start sites, and within promoter regions, defined as plus or minus 2 KB. And then one could add other annotations they're interested in either before they start the model learning, or you can do this after you've learned the model, still analyze it with other enrichments using a command I'll show you. And then similar to before, you can also download this image as an SVG file or in tab-delimited text file as well. Another type of plot that ChromHMM can automatically make is neighborhood enrichment plots. So in this case, you can see how the states are relative to a set of anchor positions. So in this case, it's showing you relative to transcription start sites, what's the relative enrichment of each state as you sort of look around the transcription start site. So it's showing you a greater enrichment in state 2, and then to some extent state 1 right around the start of genes. And as before, you can download it as SVG or tab delimited text file. What I was showing you before was all based on GM 12878, but it keeps on adding these enrichment plots for all the cell types for which the model was learned based off of. And now I'm going to give you an overview of some of the additional commands. There's a few I won't talk about, but you can read about all of them in the manual, and then I'm not going to talk about all the options. But one is the compare models command. So you could be in this situation where you have a number of different models with different numbers of states. And one way to sort of compare them is in terms of the mission parameters. In particular, you can take like one of the largest models you've learned, and then you can ask, to what extent do you have some state in models of smaller numbers of states that correlate with that state? So in this case, what I'm showing you here is, in this case, there's some model that we're comparing to that had 51 states. So those are the rows. Each one of these rows corresponds to a state of the 51 state model. And then each column corresponds to one of the models with a fewer number of states. And then darker means there's a state of this model with a smaller number of states, which is heavily correlated in terms of the mission parameter vectors with the state of this 51 state. You can go into that model and ask, what is this state here where we're not recovering it unless we have this number of states? And then if there's some sort of clear biological interpretation that's of interest, then you might want to justify, I need to have a model with at least this number of states. So that's one way to sort of get some comparison between models with different number of states. Sometimes you can also just get into a certain range that is reasonable and look at the mission parameters for each of those states. Yeah.
For different model, you mean you input different data?
So these are the same data, same options. The only thing we're changing is the number of states. So that one parameter, we're varying, and we're asking to what extent can we recover the sort of the states in terms of the mission parameters with these smaller number of state models. So make browser file. So if you have one of the model, or if you have a segmentation file, so this just works off of the bed, 4-column bed file. You can give each state, there's a color mapping file which is described in the manual, and you can also give labels, names of the states, and then it can regenerate those browser files for you based on a new coloring scheme if you desire, or giving actual state labels. And so it works similar to before, but in terms of the command line, but now you're specifying the browser file. And in general, when you're reading the manual, these things in the brackets here are optional parameters, and then these are required parameters, and they're all described in the manual. Overlap enrichment is another command. So after you've have a segmentation, you can recompute the enrichments. So you might have learned a model and then decided that you want to overlap it with some other coordinate. And you don't want to necessarily relearn the whole model using the learn model command because that can be time consuming. So what you can do is just take the, that 4-column bed file and then a directory of the coordinates you're interested in overlapping, and it can regenerate those heat maps and also output it as text file for you. And you can do something similar with the neighborhood enrichment plot as well. You can decide later that you have a segmentation in terms of the bed files and that you want to recompute some of these based on a new set of positions without relearning the model. And then there's another command, reorder the model. If for some reason you wanted to reorder the states, you need to take the original model, and then it can re-output the model with the states reordered, and then you would need to rerun the sort of make segmentation. I don't think I put that command here, but it's described in the manual as well. But you can reorder the states without, as you specify. So that sort of concludes the what I had prepared here. I want to acknowledge, so a lot of this work had been done while I was at MIT, and these were some of the collaborators on various aspects of this involved in, and a lot of this work was done in collaboration with the ENCODE consortium or the Roadmap Epigenomics consortium. In particular, I worked with the first part on with the ENCODE production group at the Broad Institute in MGH. Yes?
So the question that I have is, like, do you have any easy way to annotate the states besides getting really familiar with your markers and see what corresponds?
Yeah, so the question, so I mean, there's this manual step here that these states are, it's sort of semi-automated in the sense that the states are learned automatically, but then the human is giving some interpretation to these states. And so in order to give the interpretations, I mean, to some extent you look at what marks are associated with it, but then doing the enrichments. So, I mean, there's general things where, like, if you see a strong enrichment for promoter regions, you can classify states as sort of being promoter associated. If there's a strong enrichment for gene bodies, you can classify it as transcribed. If you're going to classify something as a candidate enhancer, you would want to see enrichment for transcription factor binding, potentially. So then, but I mean, one thing to keep in mind is not to like overinterpret these annotations. It's just in some sense a mnemonic to help sort of give some connection to the biology, but it's just sort of a human interpretation. Yeah.
How much is the window selection, or how much the enrichment sensitive to the window selection?
The 200 base pair window?
Base pair window.
I don't think it's that sensitive. With the enrichments, there's also an option to do it at the base level, or you can do it at the bin level, but they tend to give similar results.
Yeah, coming to the use of these chromatin states. So in your talk, you focused on annotating gWAS hits. But what about if you have DNA methylation data? For example, you find an result from an EBOS study, so CPGs that are associated with the phenotype. How could one tweak these tools to annotate them?
It's actually, I've been a co-author on a paper. It was published in Nature Neuroscience by Phil DeJaggers and others, which they did use these chromatin states to interpret EWAS data. So it's already a published example, and it wasn't really anything different per se. I mean, you need to be careful with the like if you're working off of array data, what the enrichment of the array was for these states, because your background's not the whole genome, it's now the probes on the array as well. But you, to some extent, you can do an overlap with that, and you would want to potentially focus on the cell types that might be matching the where the DNA methylation was conducted, if that's available.
Another question is, like, do you have any tool to compare change of chromatin states explicitly between two cell types and with the statistical significance associated with it? So people can go through, like, lists and see what is the most dramatic changes between two cell types.
I don't have one explicit way. I mean, I've looked at this problem to some extent. I mean, there is, I mean, to say, sort of the issue with most dramatic, there's, when you're working with multiple marks, now there's different ways of defining sort of dramatic, and sort of depends on what notion you're defining dramatic per se. So. Yeah, I mean, yeah, I mean, so it's the type of thing where, like, it could be the case that you could completely lose a transcription mark, but the enhancer marks are unchanged. So if you care about transcription marks, that would be a big change. But if you just care about enhancer changes, it's not a big change. So the way I've been looking at it is you probably need to have some sort of supervised guidance often in what you're interested. There is other work that's been published by other people on sort of doing differential analysis in terms of like PCA dimensions in more of an unsupervised way where they sort of figure out on what PC components are changing between samples.
I'm thinking more indirectly in your program states that, for example, you have the same number of states because you have run this, you have concatenate all the tissue samples already, and then you have the same number of states, and you have posterior probability of all states in different tissues. Can you use those for direct comparison?
Yeah, there's actually work, it was used part of the roadmap paper by Angela Yen and Manolis Kellis. I think there'll be a paper coming out at some point where if you have the data across enough cell types, then you can ask, like, if you have some region, for example, you can ask, do we see more of this state in this region in this group of cell types versus some other group? So if you have enough cell types, then you can ask those types of questions in sort of a formal statistical way as well.
Has anyone begun to incorporate the histone marks into network analysis?
Yeah, so I didn't get into this, but by analyzing the places where you have the enhancers, for example, then what you can do is you can search for regulatory motifs, for example, and that can implicate transcription factors targeting the enhancer. So that's sort of upstream to this enhancer thing. And then the other thing you have is if you have some distal regulatory element, you want to know what genes are potentially regulating. And I've done some work, and others have, where what you do is you can correlate the histone mark or the chromatin states across cell types with the expression. So if you see in a subset of cell types, you have the enhancer and another set where the gene's expressed, that gives you some statistical evidence that there's a connection between potentially this regulatory element and that gene. So that's sort of another way in sort of the network sort of connection framework that these can be used as well.